Hello dear students, welcome back again to Chinmoy's Biology channel. So today we are here with the third part of fundamental unit of life. So today we are going to discuss about the nucleus, cytoplasm and other cell organelles. So in the previous episode I have already discussed about the plasma membrane in detail. So if you have not yet gone through that part, I have given the link in the I link above as well as in the description box below. So please go through it first so that it will make your learning process easier. So now let's get started with today's episode of this nucleus, cytoplasm and cell organelles. So here we come with today's episode, nucleus. So nucleus, what is it? So remember the temporary mount of the onion peel we prepared. So in the first episode I have told you that the onion peel mount we have done in class. So that if you remember, you will see that we have seen a black dot-like substance inside the cell. So we had put an iodine solution on the peel. Why? So that what would we see if we tried to observe the peel without putting the iodine solution? Now try it and see what is the difference. Now further, if we are not staining it or we are not putting iodine solution, we will not see the features properly, right? When we put iodine solution on the peel, did each cell get evenly colored? So these are all the observations you should remember in your mind, and you should be clear that when you are putting iodine solution, all the cells were uniformly colored? No. Why? So according to their chemical composition, different regions of the cell get colored differentially. Okay, so there is a difference in the colors because the cell composition is different in different cells, and some regions appear darker while other regions less darker. Apart from the iodine solution, we would also use saffron in solution or methylene blue solution. So these are also commonly used, methylene blue solution or saffron in lab. When you go, you just ask your teacher to show you which one is the saffron in solution, which one is methylene blue solution, and which one is the iodine solution. So we observe the cells from an onion, and let us now observe cells from our own body. So what we will see now, we will do an activity, right? So this is activity 5.7. So activity 5.6 we have discussed earlier. Now this is activity 5.7. Now let us take a glass slice. So the process will be similar. So that we did on onion cells, this we will do on our own body cells. How? Now using an ice cream spoon, gently scrape the inside surface of your cheek. So pull your lip and just pull it outside, and inside of that, the inside surface, you just scrape it with an ice cream stick or a spoon. With the help of a needle, we then transfer the material and spread it evenly onto the glass slide. We are spreading it evenly to the glass slide, and to color the material, we can put a drop of methylene blue solution on it, right? So just we are scraping with the help of a ice cream stick, we are putting with the help of a needle on the glass slide, and we are now putting a drop of methylene blue, and we should never forget to put a cover slip on it. With the help of a needle only, properly place the cover slip so that no air bubble should enter inside that, right? So the same procedure as we have done in the previous one. So what do we observe? What is this shape of the cells we see, and draw it in a sheet of paper. So you are doing this, you are drawing it on a sheet of paper, and we are writing the observation. So was there a darkly colored spherical or oval dot-like structure near the center of each cell? This structure is called the nucleus. So same nucleus we have observed in the onion peel cells, and this we observed in our own cheek cells, that is our own animal cells also. So this is the nucleus. So what is nucleus now? Now we will discuss it in detail. So nucleus, it is a double layered covering called the nuclear membrane. The nuclear membrane has pores which allow the transfer of material from inside the nucleus to its outside. So the same way as the transfer of materials were happening within the plasma membrane, that is inside the cell and outside the cell, from the nucleus also the materials come inside and outside the nucleus to the cytoplasm, which we will be talking in the next section. Okay, now what does the nucleus contain? This is very important. The nucleus contains the chromosomes, which are visible as rod-shaped structures only when the cell is about to divide. So When we will study about cell division, we will understand that when the cells are about to divide, we will see this rod-shaped structures which are called the chromosomes. Now, the chromosomes contain information for the inheritance of characters from the parents to the next generation in the form of DNA, that is deoxyribonucleic acid. So, deoxyribonucleic acid is the, we can say that this DNA is helping to inherit the characters from the parents to the next generation in the form of DNA, so that is deoxyribonucleic acid molecules. Okay. Now, chromosomes are composed of DNA and protein. So, what are chromosomes composed of? They are composed of DNA and protein. So, DNA molecules, they contain information necessary for constructing and organizing the cells, right? So, DNA molecules contain information necessary for constructing and organizing the cells. Now, functional segments of DNA are called genes. So, some new things you are studying today. First, you studied chromosomes. Then you studied about DNA or deoxyribonucleic acid. Then we have studied about the functional segments of DNA that are called genes. Okay, genes. Now, in a cell which is not dividing, this DNA is present as a part of chromatin material. So, this is another new term, that is chromatin material. If the cell is dividing, you will see then it is in the form of genes. The DNA material is inherited or transmitted from the parent to the offspring. Now, if it is not dividing, the DNA is present in the form of chromatin material. Now, chromatin material is visible as entangled mass of thread-like structure. So, during cell division only, these are visible as entangled structures, and whenever the cell is about to divide, the chromatin material gets organized into chromosomes, right? So, what is happening here? We will go to details again with the diagram. So, the nucleus is playing a very crucial role in the cellular reproduction, right? So, the process by which a single cell divides and forms two new cells. So, nucleus contains the nuclear material, it contains the chromosomes, it contains the DNA or the chromatin fibers, which are all genetic materials and which are helping in the cell division, right? So, it is also playing a crucial part along with the environment in determining the way the cell will develop and will form it exhibit at the maturity by directing the chemical activities of the cell, right? So, it is actually having a very important or crucial role in the process of cell division. So, is it clear to everyone? Now, we will move on to certain organisms like bacteria whose nuclear region is not well developed, okay? It may be poorly developed due to absence of the nuclear membrane. So, they do not have any specific nuclear membrane to cover the nuclear material. So, it is not well developed. So, that is called nucleoid. So, that is not nucleus, that is nucleoid. So, such organisms or cells lack a nuclear membrane are the prokaryotes. So, prokaryotes means, pro means primitive or primary, and karyot or karyon means the nucleus. So, it is a primitive kind of nucleus, right? Now, organisms with cells having a nuclear membrane are called eukaryotes. So, those who are not having the cell membrane are called prokaryotes, and those who are having the cell membrane, they are called obviously the eukaryotes, right? Now, prokaryotic cells also lack most other cytoplasmic organelles also, right? So, these are not but they are all present in the eukaryotic cells. So, many of the functions of such organelles are also performed very poorly organized parts of the cytoplasm. So, the chlorophyll in photosynthetic prokaryotic bacteria is associated with membrane vesicles, that is bag-like structures, but not with the plastids as in eukaryotic cells. So, in plastids, the chloroplasts are associated with plastids in eukaryotic cell, but in the prokaryotic cell, they are associated with certain membranous vesicles. So, let us see the cell structure now. Now, this is a prokaryotic cell. This is having a cell wall. This is, we can say that this is the plasma membrane. And this is the nucleoid region. So, this is some cell organelles inside the cell we can't make out what are these because they are not fully developed. This is the nucleoid. This is not having a nuclear membrane. These are the ribosomes. And this is the plasma membrane. So this is a prokaryotic cell. Now we will see in the next slide the prokaryotic cell, the eukaryotic cell, and we will put a stress on those chromatin fibers, DNA and all. Let us see that. Then we will move on to the cytoplasm. Now you can see here 3 diagrams I have shown you. The first one is of a prokaryotic cell. So prokaryotic cell, here you can see the nucleus material. They are not covered by a nuclear membrane, right? So that part is called the nucleoid. This is the outer capsule. This is the ribosome part. This is the cell membrane or the plasma membrane. This is the cell wall. These are the plasmids. And we can say that this is the cytoplasm, and inside the cytoplasm only the DNA material of the nucleoid is spread. It is not in a compact form as it is in nucleus of a eukaryotic cell. So this in the right side, this is a eukaryotic cell. So in this cell you can see the nucleus is covered by a nuclear membrane, and inside the nucleus there is again a round ball shaped structure which is called the nucleolus. So nucleus, nucleolus, and the nuclear is covered by a nuclear membrane. Now this outside the cell is a cell membrane. This is mitochondria, these are ribosomes, these are endoplasmic reticulum, and these are Golgi apparatus. So these are all the cell organelles inside the cell. We will be discussing it now itself. Now just for your clear vision of this prokaryotic and eukaryotic cell structure, I have taken this diagram. Now we will see that what are chromosomes and how do they look like. So inside the nucleus of a cell, chromosomes like this, they are present, and these chromosomes, they are having this double helical structure with ATGC base pairing. So this ATGC base pairing, you will be studying about this in higher classes, not in 9th standard. So let's move on to the next part, that is the nucleus. After nucleus we will move on to the cytoplasm. Now we are here discussing about the cytoplasm. So when we are looking at the temporary mound from an onion peel as well as human cheek cells, we can see a large section of each cell enclosed by the cell membrane, right? So this large region which is enclosed by a cell membrane which takes up very little stain, we can say that it is almost white in color only. That little stain taking part is the cytoplasm. The cytoplasm is a fluid content inside the plasma membrane. It also contains many specialized cell organelles, right? So it is containing many specialized cell organelles. Each of these organelles perform a specific function for the cell. So there are many cell organelles. So cell organelles in detail we will be studying, but not today. Today already we have studied a lot. We have studied regarding the plasma membrane. We have studied a lot regarding the nucleus. We have studied prokaryotic, eukaryotic cell, chromosome, chromatic fibers, DNA, gene, and also those new things we will be taking them in the next episode. So cell organelles are enclosed by membranes. In prokaryotes, beside the absence of a well-defined nuclear region, the membrane-bound cell organelles are also absent, right? Now in prokaryotic cells, we have told that they don't have membrane-bound developed cell organelles, so that is absent in the prokaryotic cells. On the other hand, in the eukaryotic cells, we have nuclear membrane as well as all the membrane-bound organelles, right? So the significance of the membranes can be illustrated with examples of viruses. Viruses lack any membranes, hence they do not show characteristics of life until they enter a living body and use its cell machinery to multiply. Right, is it clear? So until and unless they are using their cell machinery to multiply, they lack if they are lacking any membranes, and hence they do not show any characteristics of life until they are entering the host body or the living body, right? So they are lacking all the activities of a living body until they are entering the host body. So there only they can multiply. Now here, fill in the gaps in the following table illustrating the differences between the prokaryotic and eukaryotic cells. So they have given you the differences, just you need to fill in a few, right? Now for the prokaryotic cell, the first one is regarding the size, generally small. So this and this they have given the answer in full. So then from the nuclear region you need to write. In the eukaryotic they have written well defined surrounded by nuclear membrane. So it is not well defined, and what is it called? It is called nucleoid. Just now I have discussed it. Now chromosome is single in prokaryotic cell. So more than one chromosome is there in eukaryotic cell. Now in prokaryotic cell, membrane bound cell organelles are absent, whereas in eukaryotic cell, membrane bound cell organelles are present, right? So here the membrane bound cell organelles are present in the eukaryotic cell. So this is what is the difference between the prokaryotic and the eukaryotic cell. Now we will move on to the cell organelles in details in the next class itself. So in the next class, we will go to the cell organelles in details with all the things like endoplasmic reticulum, where we will be studying about smooth endoplasmic reticulum, rough endoplasmic reticulum. Then we will understand about what is Golgi apparatus, what is their function, what are lysosomes, and what is mitochondria and plastids. Vacuoles also will be mentioned in the next class. Then we will see how the cell division is happening, the process of cell division, and also in the next 2 classes we will complete this full lesson, because this is a vast lesson and I am dividing it into different parts so that it is easier for you to understand. If I make you understand the full lesson in one episode itself, it will be very difficult for you. So you need to subscribe to my channel if you want to know more regarding this lesson, that is a fundamental unit of life. In the next class, I'll be teaching you something about cell organelles, and after that, I'll be teaching about the cell division. So stay tuned, stay updated, and stay with all my lessons which I am updating. So till then, complete whatever I have taught in today's episode as well as in the previous 2 episodes. If you have not yet referred to that, I'll be putting the link in the description box. Okay, thank you.